the City University, Professor Kabir Adrian. We are also fortunate to have in our midst our own father, the Chancellor of the City University. We all know our Chancellor is always supportive of everything we do here. He's the captain of the training of scholars on this campus. I'm talking about Professor Umali. Permit me to invite the Vice Chancellor for his opening remarks. Let's put our hands together.
Our indefatigable Chancellor, Professor Gabriel Ogunmola, Fellow Academy of Science, the Registrar, Dr. Bola, our guest speaker for today, Professor Vincent C.O, Njar, that is a PhD, University College London. Let's give me a round of applause. Deputy Vice Chancellor, Dean, Professor Shilala, the Provost College of Medicine, other principal officers of the university, Dean of Faculties, the University Librarian, I recite you. The distinguished and our esteemed staff and students, please give yourself a round of applause. We thank you, despite the fact that we are on vacation, you still find time to be here. We are very proud of you. Compliment of the season to you all. I'm delighted to formally welcome you all to the 2024 First Distinguished Lecture, an event that underscored university commitments to fostering a culture of academic excellence, exploration, and discovery. As we all know that a distinguished lecture is a presentation given by an eminent and accomplished individual in a specific field to provide valuable insight and expertise to a broader audience. And we are lucky to have today, at the heart of today's gathering, the distinguished speaker, that's Professor Vincent C. Njar, a luminary whose credentials speak volumes about his contribution to the field of medicinal chemistry and pharmacology. Having earned his PhD from University College London, Professor Njar brings a unique blend of academic rigor and inventive spirit to our university. Once again, on behalf of the Chancellor, the Council, and the top management of Blissett University, I welcome you to our campus. The theme of today's lecture is a journey in academic medicinal chemistry, biomolecular chemistry, and drug discovery, ingenuity and serendipity, or is it in the other way around, which I believe will capture the essence of the challenges and triumphs inherent in the pursuit of cutting-edge knowledge and research. The focus on biomolecular therapeutics approaches in prostate, pancreatic, and breast cancer is a particular project considering the global impact of this disease on public health. Our distinguished scholar today promised to give us an insight to unravel the complexity of these ailments and present potential avenues for therapeutic interventions underlining the crucial role that academic research plays in addressing real-world challenges. We are very proud to have you. The very nature of academic research is often characterized by delicate dance between ingenuity and serendipity. As we delve into the dynamics of medicinal chemistry and drug discovery, this introspective exploration promises to challenge our preconceptions and expand our understanding of the specific method. For those who are unable to join us physically, we have embraced technology to embrace an online platform participations via event lcu.edu.ng, which I think our online participants will be able to join us. This inclusivity ensures that the knowledge and insight shared today will transcend physical boundaries, reaching a global audience and fostering collaboration beyond our immediate surroundings. Once again, I extend my warm welcome to our esteemed guests, I believe that with your coming to our campus today, this will be the beginning of collaboration, partnership, linkages, and symbiotic relationship has been initiated when we paid a visit call to my office, and the Chancellor was able to have a brainstorming event with you. I would like to appreciate the DBC, Professor Ladogo, and our team for a wonderful job well done. Once again, ladies and gentlemen, on behalf of the Chancellor, the Council, and top management of Vincent University, I welcome you to this distinguished lecture. Let us approach this distinguished lecture with open minds and eager hearts, ready to absorb the wealth of knowledge that our guest lecturer has to share with us. May the discussion that followed be characterized by intellectual curiosity, sparkling collaboration that will propel this university to a new height of academic distinction. Distinguished ladies and gentlemen, I welcome you to this program. Thank you.
Thank you, sir. Permit me to stand by the already established protocols by the Vice Chancellor, while I ask Professor Njar to please rise while I take his citation. Professor Vincent Njar completed his BSc in Chemistry at the University of Ibadan, the top university in Nigeria, in 1976, and went on to obtain a PhD in Organic Chemistry in 1980 from University College London, University of London, UK, under the supervision of Derek V. Banthorpe. Following 2 years of postdoctoral research with the late Elihu Caspi of Worcester Foundation for Experimental Biology in USA, he joined the Department of Chemistry at the University of Ibadan, Nigeria, as Lecturer 2 in October 1982. He was promoted through the ranks and became Professor of Organic Chemistry in 1996. Professor Njar is a recipient of numerous awards, including Fogati International Fellowship Award, 1986-1987, Yamagiwa Yoshida Memorial International Cancer Award, 1989, Medical Research Council of Canada Award, 1990, International Cancer Technology Transfer Award, 1992, and an Alexander von Humboldt Research Fellowship Award, 1994 to 1995. During his tenure at University of Ibadan, he was visiting professor at several institutions, including Johns Hopkins University, Baltimore, USA, 1988, 1989, 1992, and 1993. University de Sherbrooke, Quebec, Canada, 1990, University of Southampton, UK, 1991, 1992, and 1995, and University of Saarbrücken, Germany, 1994 to 1995. Following several research visits from 1996 to 1998, collaborating with the late Angela Brody at the University of Maryland School of Medicine, Baltimore, internationally renowned breast and prostate cancer researcher, he immigrated to the US in 1999. He is currently Professor of Medicinal Chemistry and Pharmacology, Department of Pharmacology, and Head Medicinal Chemistry Section of Center for Biomolecular Therapeutics, CBT, University of Maryland School of Medicine, Baltimore, MD, USA. He's also one of the 5 inaugural Distinguished University Professors, UNB's IS Faculty Honor. His main research interest is in the design, synthesis, discovery, and development of novel small molecules as therapeutics for the treatment of human cancers, particularly prostate, breast, and pancreas, and other diseases. Over several years, he has developed a comprehensive drug discovery and development program at the University of Maryland School of Medicine. His research group consists of 2 laboratories. His medicinal chemistry laboratory has expertise and capability in target identification, rational design of compounds, molecular modeling, lead identification, and lead optimization using modern synthetic medicinal chemistry methods. His cancer, molecular biology, and pharmacological laboratory has expertise and capability in assay development, molecular mechanistic studies, in vitro and in vivo pharmacokinetic, toxicity, and anti-tumor, anti-metastasis efficacy studies. In collaboration with Dr. Angela Brody, they invented a novel CYP17 inhibitor, AR antagonist androgen receptor degrader, ARD, VN1241, now named Galateron, that advanced to pivotal phase 3 clinical trials in ARV7 positive metastatic castration-resistant prostate cancer patients. Although the trial was unsuccessful, the Galateron technology has been licensed to LCN Pharmaceuticals Incorporated, Baltimore, that is continuing its clinical development. LCN will soon initiate a global pivotal phase 3 study of Galateron in metastatic castration-resistant prostate cancer patients. The trial is programmed to start enrollment of patients in Jamaica during Q3 and Q4 of 2023, and in 4 African countries in Q1 of 2024. Additionally, Galateron is in phase 2 clinical trials in patients with metastatic pancreatic cancer. The next generation Galateron analogs, NGGAs, are also in development. Let me just quickly skip to some of the important parts. Current projects in this laboratory include 1. Development of next generation Galateron analogs, NGGAs, also called GalNex, novel and efficacious androgen receptor degraders, ARDs, and MNK12 degraders, MNKDs, to target prostate and pancreatic cancer growth and metastasis inhibition. And 2. Development novel retinamides, NRs, as new therapeutic agents against breast and prostate cancers. Professor Njar has published 123 peer-reviewed articles, reviews, and book chapters, and is the lead inventor of 37 issued US foreign patents and more than 30 pending patents. And more than 30 pending patents that have led to several highly productive increasing agreements at UNB. On 85% of these, he is first or senior corresponding author. The impact of his research has an age index of 47 and 19,499 citations as of October 11, 2022. Professor Njar actively participates in several drug discovery and development grant review committees, nationally and internationally. His research is currently supported by the US National Institute of Health, NIH, and the National Cancer Institute, NCR. His inventions/patents have been licensed to biotechnology companies including Tokai Pharmaceutical Incorporated, LTN Pharmaceuticals Incorporated, Baltimore, and Prof. Therapeutics Incorporated. Indeed, the impact of Dr. Njar's research and inventions and patents were recently, in 2021, recognized by UMB's president, Bruce L. Gerald, MD, FACS, as one of the 5 University of Maryland professors recognized with the inaugural designation of Distinguished University Professor. The title of Distinguished University Professor is the highest appointment bestowed on a faculty member at UMB. It is a recognition not just of excellence, but also of impact and significant contribution to the nominee's field, knowledge, profession, and practice. Furthermore, because of his excellence in entrepreneurial activities, UMB elected him as David J. Ramsey Entrepreneur of the Year 2021. The 2021 National Academy of Inventors, NAI, Fellow Selection Committee and Board of Directors elected Professor Vincent Njar to the rank of NAI Fellow, FNAI, He was inducted at the Fellows Induction Ceremony at the NAI annual meeting in June 2022 in Phoenix, Arizona. Professor Vincent Njar has over 40 years of demonstrated accomplishments as a medicinal chemist and oncopharmacologist in academia at several universities, including 25 years with the University of Maryland School of Medicine, Baltimore. He has over 120 scientific publications, 37 issued patents, and 30 plus pending patents. H-index 47, citations 19,454 as of 25th of September 2023. He has a PhD in chemistry, University College London, and completed a postdoctoral fellowship at the Worcester Foundation for Experimental Biology in USA. Ladies and gentlemen, permit me to present to you today our lecturer for the Distinguished Lecture 2024, Professor Vincent C.O.
You have heard almost all about me, because I hope I'm not going to disappoint you in the presentation of my talk. So that's the challenge. Good afternoon, everybody. I am, this very pleased. To be here with all of you today. And some of you in person and others online, virtually. To listen, to my talk as part of LCU's distinguished this lectureship series. I am humbled and honored and grateful for this invitation. I thank the chief host, that's the Vice Chancellor of this great university, the Deputy Vice Chancellor, who I've been dealing with over the last couple of months in arrangements for this talk, and also the registrar here seated. And finally, for those I have to thank, your Chancellor, that's Professor GP, and I would like to tell you he was my former teacher. He was my former teacher as a brother. I am aware that the major mission of the University, to further rigorous education, through various ways, including this distinguished lectureship series. Ladies and gentlemen, I would have loved to talk about your distinguished However, I do not have the time to do so today. So I would actually proceed and to discuss today's lecture. I mean, in preparing this lecture, I took into a serious consideration the diverse backgrounds of the audience. However, to appreciate my talk more fully, one needs to understand, that's the rudiments, of medicinal chemistry, oncology, molecules, drugs, and the language is there. However, I will try to emulate that Einstein's V2, which says we should make things as simple as possible, but not simpler. So, I will now go ahead to start my lecture. And the subtitle of my lecture is Medicinal Chemistry Drug Discovery. Is Ingenuity and Serendipity, or is it the other way around? And that's big academia. That Leeds City University. It's an academic institution. My talk will be divided into two parts. First, I will discuss whether prostate and pancreatic cancers. The rationale for the development of our lead compound with Ingenuity for prostate cancer. The current status, and I'll also discuss that's the clinical trials that are ongoing at our university in this area. And I'll also discuss that's the next generation skeleton analogs. In the second part of my talk, I'll discuss about breast cancer, highlighting that we run by these molecules. I'll just summarize the key data for lead compound, deuterated analogs, the company in which I formed with University of Maryland, Baltimore. As required by the University of Maryland, Baltimore, I have to make these disclosures. VN 124.1, top 001, now called skeleton, was discovered or invented in my laboratory. That's in collaboration with the late Angela Crowley shown here. And this technology was licensed by UMB, that's to talk about biopharmaceutical for clinical development. And as you hear, it's now licensed to another company, this LTN Pharmaceuticals. Here, this was the ceremony of the signing of the licensing to ESL, now LTN. That's the university president, that's the former president. He's now the chancellor of the University of Maryland system. That's Dr. Perman. That's the head of our office, of technology transfer. That's the president and head of LTN. That's the CEO. So, in licensing technologies, the university, this leadership is fully involved in all these processes. So this slide, highlights, that's the drug discovery from academia. Academic research. Some drug discovery program research are anything but academic. All of us know, the word research means a careful and diligent search. It's a studious inquiry or examination. Very nice, noble activity. Okay, but academic is a horse of a different color. In its now form, it means a person who is very learned but inexperienced in practical matters. Adjectival form, having no practical or useful significance. And as a synonym, is pedantic. Usually, when you say academic, it means it does not really matter. However, Mr. Vice Chancellor, I want to do things that matter. I certainly would not want to be associated with the term like pedantic. This brings us, to an important point. If we agree, with this definition, that's a dictionary definition of academic, it means that all of us here, are wasting our time. We're not doing things that matter. So I believe that, we should try to change, that's the definition of academic. This slide here shows the overall strategy of research in my lab. The medicinal and organic chemistry lab, this target identification, the rational design of compound synthesis and molecular modeling. The preclinical studies, the pharmacology and oncology, that was the in vitro work, the in vivo work. And if you train in my lab, you would train in all these areas, organic chemistry, medicinal chemistry, and so on. And the goal of my lab is to be able to move things from the bedside, clinical trials, and eventually, have new drugs for humanity. This slide here, just summarizes what we do. You have to come up with an idea, that's a hypothesis. And once you have a hypothesis, you have to have funding. Do your research, is shown here, this in vitro. And when you get a lead compound, you can move that now to study in mice. That's preclinical studies. And then with the clinical, the studies or trials, there are three major phases. The phase 1, you want to test whether these molecules are safe in humans. You want to also find out the dose that you will use in your phase 2 studies. And from the phase 2 studies, if it's successful, you advance now to the phase 3. That's randomized, it's trial, and you compare with existing drugs. However, because of lack of time, I will just summarize what we all started out. We started out, we tried to develop an enzyme to inhibit the CYP17. And the idea is that if you inhibit this enzyme, the androgens will be decreased, because it is known that androgens would stimulate the growth of prostate cancer. And if you inhibit the CYP17 enzyme, which is critical, as I will show in the next few slides, you will now reduce the levels of androgens, that's the castrate level, and the tumors will regress. We now know that the major source of androgens in our bodies are, that's from the testes, that's the major source, over 80%, from the adrenal gland. And of late, we have found out that the prostate cancer cells themselves are able to produce these androgens. This here shows the male reproductive, these organs. That's the prostate gland below, that's the bladder, and it surrounds, that's the urethra. Charles Higgins, won the Nobel Prize in Physiology and Medicine in 1996 for the invention of castration as a therapy, for prostate cancer. So what he found out was that, if you castrate the patients, you reduce, that's the major source of androgens, and that was what he got the Nobel Prize for in 1996. As an alternative, that's the castration, we became interested in developing the key enzyme that the CYP17, which I will show in the next few slides. In prostate cancer, we start out as a small tumor in the prostate, and it can now advance, it gets enlarged. However, what kills the patient is when it spreads to other organs of the body, like the bones and the lungs. And it is shown that the 5-year survival rate for prostate cancer that is spread to other organs is 28%, while it's almost 100% when it's not spread to other organs. Okay, so what happens when you inhibit CYP17 enzyme? So if you see this pathway starts out, that's from cholesterol, this pathway. And the CYP17 enzyme operates in 2 steps, the hydroxylase and the lyase step. So what happens when you inhibit CYP17? There's a downregulation of testosterone and this PhD. I'd like to highlight on this slide that, in Nigeria especially, you are being told, that cholesterol is a very bad thing. Ladies and gentlemen, if you do not have cholesterol in our bodies, you would not have the male and the female sex hormones. Remember, the androgens are called the male sex hormones. And the androgens are the ones, which are transformed in one step, to the female hormone, estrogens. So you should not think that, this cholesterol is that bad. If you don't have it, you are not the male or female. Okay, how do these androgens work? They have to bind, that's the receptor. That's the androgen, this receptor. This here is the full length. And during, that's COVID-19, you heard of, that's the splice variants. Androgens also have, yes, splice variants. And this variant is also functional in the system. So how does this work? The androgens, that's testosterone, gets converted to the more potent androgen DHT, shown in this reaction. They bind to the androgen receptor, there's phosphorylation, and the dimerize, and they bind to the response element. And this then will make the prostate cancer to grow. And all we try to do is to inhibit that the formation of this. Trying to block that androgen production is currently the mainstay therapy for prostate cancer. So I told you our goal was to design and synthesize compound that inhibit the key enzyme CYP17, which I've shown you the pathways. Our strategy for our design was based on the knowledge of the active site of the enzyme, which we know has a hydrophobic region and a heme prostatic group. And also the mechanism of the reaction, starting out from pregnenolone or that's progesterone. There are 2 main steps. So that's how we designed our molecules. The crystal structure at the time wasn't known. So if you look at this molecule here, that's pregnenolone, we then reasoned out that if we can modify this region of the substrate for the enzyme, we may then get a molecule which would bind, of course, to this hydrophobic region, and with the heme ion will form a coordinate bond to the heme ion, and inhibit the reaction. Remember, this heme ion, would activate, that oxygen which is involved in this reaction. So if you block this activation step, you inhibit the reaction. Also critical to this, we invented, that's a new reaction. And how did this reaction occur? Again, you should listen to this slide properly. We actually wanted, to hydrolyze the acetate at this position of this molecule, to get this. However, we got this molecule, that the hydrolysis took place. But the chloro group at C17, got eliminated, with substitution of, that's a toxic group. We then reasoned that if you have an azo in this system, you may be able to eliminate, eliminate the chloro and the azoles. That's how this reaction was invented. At the time that we discovered this reaction, we now were able to show that our invention was the first in which an azole group is linked. That's the steroid molecule there. That's the nitrogen. And of course, we went ahead and discovered that's galactone, which I will talk about in the next couple of slides. This is the synthetic scheme that was developed in my lab. And this synthetic scheme, is currently used. That's for the large scale synthesis, the commercial scale synthesis of this molecule. Here, I'll just show you the impact of our molecule. When you want to test compounds, you try to compare them with known therapies and with known drugs. So as you can see here, that's castration is compared. But one of our lead molecules there, that galactone is quite effective. I will not go into the details. And if you look at, all these 6 studies, independent studies, our molecule, was better, than either castration or FDA approved drugs. So, trying to summarize the studies, so far, we have, the pathways which are dependent on the androgen receptor. That's also fine. But we also found out other pathways which are inhibited and which don't depend on the androgen receptor. In the second half of my talk, I'll discuss more about this protein synthetic pathway, which involves the A30, the PI3 RAS pathway. It's important, in most cancers because it is found in most diseases. So as you can see here, we just follow the science. Whatever we found, we're able to exploit. Again, Ingenuity, Serendipity, or is it the other way around? In the next couple of slides, I will discuss the clinical trials with this agent. So the phase 1 clinical trials, what you want to find out is a dose that you think is effective. That or doses which you would use in subsequent these phase 2 trials. This is a plot. That's a waterfall plot of the PSA. And the PSA, you can assay it, from the patient's blood, serum. So if you take, this point as 0, that's the starting point of the PSA. Those patients, we don't respond. It's, on the other side of this waterfall. So it was able to show that when we're able to show, of course, the galactone was effective, and then it was now designated fast track, and then the phase 2 started. But if you notice here, the dose of galactone was quite high. In the next few slides, we'll discuss the impacts of this. We have in 4 distinct, these populations. In a study where you have, the treatment naive and also refractory, to known FDA approved drugs. These are the endpoints. And as you can see in this slide, as most patients in this phase 2 study, did respond. So the PSA on this side means, they actually responded as to galaforum, just compared here. And just a few patients did not respond. At the time we're doing our studies, this molecule, that's Zytiga, was approved as a drug that inhibits CYP17. Another molecule, that's enzalutamide or Stansia, was also approved for this treatment. And we're able to show, remember I told you about the splice variant androgen receptor, which are important also for the etiolation of the disease. But we find out that our molecules, as galaforum, are able to induce the degradation. It means that in patients that express the splice variant, our molecule will be effective, unlike abiraterone and enzalutamide. We were able to publish this in the journal, Medicinal Chemistry, which is the most cited journal of medicinal chemistry in the world. And based actually on the clinical data and the fact that our molecules were also degrading the splice variant androgen receptor. And so Tokai now designed the phase 3 clinical trials, trying to target the patients that express AR7. They went back to look at their phase 2 data, and of the 7 patients they identified which had the splice variant, there are 6 patients, this respond 86%. That was quite high, although the phase 2 was not planned actually to look at splice variant. But based on this information from my lab, and also the clinical data, they had a phase 2 study is comparing it to enzalutamide. However, this trial failed, did not succeed. On the next few slides, I asked the question, why did galaforum not succeed or fail in phase 3? And there were write-ups which claimed that the trial design was likely to fail even before the results were shown. But the biggest flaw was the sample size of the study. The 2 drugs that I mentioned were approved, you can see, they have this number of patients, 1,000 plus patients, while the phase 3, the galaforum, had very few patients. So we strongly believe that, the phase 3 and the failure in phase 3 is actually due to the flaws in the design strategy. Another thing that happened that we noted also was that during the planning of the phase 3, the top people at this company that was developing it, they were all selling their stocks, which is, it's quite curious to us up to today. So this slide just summarizes, all that I've said. We found out it was effective even in the cancers that had the splice variant, and of course the clinical trials and the phase 1 and 2 are very promising, failed in phase 3. So to rescue this galaforum, there's a new trial that is planned, which I will discuss in the next few slides. I just want to note that in this project, you have to have many people involved because it goes back several years. So this slide just shows the graduate students, we have postdoctoral fellows who worked on this project. So what are the plans now to rescue this galaforum? So we were able to acquire some tablets, this led from the phase 3, which was estimated to be about 3.3 million. We were able to store them for over a year. they were recertified, and we got funding actually to store them. That by David Weber, who is director of our that CBD. I already mentioned to you that Galapagos is now licensed to LTN. So LTN plans to have that's a randomized clinical trial in the Caribbean. And the PI, the principal investigator, is Arif Hussein, who is in our cancer center in Maryland. And they plan to do another phase 2. We're starting out that's in the Caribbean because if you look at that's the Caribbeans, has the highest that's mortality rate of prostate cancer. And it turns out also the CEO of LTN is from Jamaica originally. So this is a slide which shows the proposed or the ongoing clinical trial by LTN. These are the primary endpoints and secondary endpoints. And we are hoping that the trials will also expand that's to Nigeria and 4 or 5 other African countries. So this is the criteria. And from some new discoveries in my lab, concerning the Galactoran source, which I will discuss under the next generation gal hormones. LTN is planning to do another phase 2 clinical trial comparing the Galactoran source with the parent compound. That will be done in the United States. Remember, I told you we started out with the androgen receptor, and we also found out that these molecules do inhibit that's protein translational complex, which I will discuss in more detail in the second part of my talk. And it turns out that protein translational complex is upregulated in most cancers, including that's pancreatic cancer. So in pancreatic cancer, there's an upregulated, and there's a mutation, of KRAS, it's right up here. And the PI created pathway will impact protein translation. And in a clinical trial, it was found out that patients that have a high level of dysfunction of this pathway, the prognosis is very poor. That's for my patients which have low levels of this. So that's the rationale for why we're now started testing our molecules that's in prostate cancer models. So in history, of course, we got an R21 grant from the NIH. We also got funding from Turkey. And this slide just summarizes the key data that we got. This publication which was very promising. And we had the manuscript published, which says that galactoran and its analogs inhibit MNK EIF4 axis. Can be synergized with the frontline drug that's gemcitabine in prostate cancer patients. So what is the significance of these discoveries? Like I said, that gemcitabine and others are the frontline drugs. But the prognosis actually the treatment with this is very poor. So our idea is that we should be able to combine our molecules with the frontline drugs with gemcitabine. This foresight from the director of our cancer center, that's Kevin Coleman. He was able to provide funding, internal funding, for us to do the clinical trials at the University of Maryland Cancer Center. So we're able to get an FDA approval for this clinical trial, So it's a phase 2 clinical trials. If you Google that, if you Google this link, you will see more about this trial. And the PI, and the PI, Dr. Njar and other co-investigators. So this trial, this started, I believe, with us in 2021. So the recruitment was very poor. Okay, and it was a single, hospital trial. But however, we obtained an email from the medical oncologist, Dr. Njar, and he says, I would like to share good news with everyone, which I will expand here. They found out that in one arm of the treatment patients, there was, that's the partial response of the patient, that failed, all the frontline therapy. That's what prostate cancer. That was quite exciting for them and us. So our cancer center director now asked, how can we expand this to other centers? We have a budget, to expand this study. He's more excited and he wants to expand, that's the internal, clinical trials, which would not be that expensive. So, so we have a plan actually, to go ahead now and expand the pancreatic cancer clinical trials. In the part 1B, we're discussing, the next generation bilateral analogs. This grant or this project, it's funded by grants from the NIH. Now we have some supplemental funding for this grant. You want to ask the question, what is the justification for synthesis of the next generation of analogs? Remember I told you that in the phase 1 and 2, the dose of bilateral was quite high, per day. And the idea is that in the next generation compounds, it can improve, on the efficacy, the pharmaceutical, these profiles. We're able to do some studies, which is already published in German Chem, and from the structure, these activity relationships, we found out that we can improve efficacy of a molecule when we modify this region highlighted here. Now we have two lead compounds, the VN 414 and the 433. It's repeated. We have published some work on this. But the problem was, the yields to these products were very low. We have solved this problem where we have a light scale synthesis now of this molecule, which improved by about 6-fold. This slide here just shows the synthesis of these molecules, which I'm not going to discuss. That's for the sake of time. We already have applied that for patents to protect the light scale synthesis of the new analogs. And we did, the PK studies to see whether we can be able to improve. That's galateron. You can see the half-life of galateron is short, but with the new molecules that we have improved on the efficacy. If we highlight, for example, with the 433 beta, the half-life increase with that 25 times, and give this molecule, this orally, and other PK properties that also improve. So the summary is that we have quite some lead compounds which are quite good. Now we tested them also is comparing with FDA approved drugs. And our molecules are better. Then I will quickly now make salts of these molecules which I've mentioned. That's galateron. That's the monosalt shown here, the hydrochloride. And the 433, we have the mono and the hydrochloride. What you should understand is that there's most drugs that we take, almost 50% are in what you call the salt form. And the reason why you have them in the salt form is that you can take them in most cases orally, and I mean they will penetrate into the cells is much more easy. So we did make the salt forms. We started out with the HCl salt, and if you look at that's approved drugs for the salt, of drugs, you can see this HCl is very prominent right here. So I'll just go quickly. Okay, this in vitro, usually, when you make new compounds, whether they're derivatives of a known compound, you also have to make sure, they're doing what you think they're doing. So this was a screening, actually, testing, of two groups of these prostate cancer cell lines. So this one's in green are that's from white patients, while the one here in pink are from black patients. There is that's emphasis now to say, when you are developing drugs, you should try to make sure they work in black patients and also in white patients. Because a lot of the drugs that we now take in this country or in Africa, almost all the studies were done with white patients. So now there is this emphasis, that you should also test to see what they do. That's ancestors. To quickly state, I think I have that below here stated. We found out that our molecules are effective in both the cell lines is derived from white, and the cell lines is derived from black patients. So we think we're doing the right thing. Of course, when you design compounds and they inhibit the cell growth, you want to ask yourself, what about the molecular markers that you think you're doing? And you have to test that. That's Western blotting. And as you can see here, we are looking for the impact of the AR and other markers. And you can see, if you just eyeball the studies, you can see that the salts are better than the current compound. So we're so excited and asked the question, how would these molecules perform in vivo? Remember, you can do the in vitro studies. If they don't work in animal models, it doesn't mean anything. All right. So we did a pilot study, and you can see, that's the control, with the salts that we had. You can see the salts are quite good in inhibiting the cell. These are the pictures actually at the end of the tumors. That's the control tumors, and you can see that the salts are quite effective. So if you look at this, I'll just highlight this. With the gal salt, we're able to improve the efficacy by almost tenfold. With the 433 salt, we were able to improve the efficacy, just by making the HCl salt by 4.3-fold. This point is important. You're looking at the body weights of the mice. If you have a drug, you want to make sure it is safe. So the easiest way to check whether your molecule is safe is to look at their body weights during the studies. If the body weights fall, it means that your molecule is toxic to the mice. And if it's toxic to the mice, all this don't matter, because you're not able to develop it at all if it's toxic. So we did a pilot study, we're excited. So the next study is we now compare with FDA approved drug for prostate cancer. That's enzalutamide. That's docetaxel. And as you can see from these curves, our molecules are much better. There's tumor regression actually to those molecules. These are, the individual solid tumors. So if you compare these tumors in this group, and these are FDA-approved drugs, which are being sold currently all over the world. As you can see in these studies, we have been able to develop molecules that are better than FDA-approved drugs, at least in these models. With the 433 also, the same thing. And we have molecules that are better than FDA-approved drugs. Okay, so this slide just summarizes the tumors. Okay, that's at the end of the study. And we excise the tumors and we photograph them. So if you look at this, those are the FDA-approved drugs. It's drugs, with this slide. That's Galateron and the salts. Look at different doses. That is the 433 and different doses. Okay, that's the control. Again, we look at the molecular biomarkers, and as you can see, our molecules are much better. If we want to summarize this section, then we have lead compounds which do what we want. Downregulate AR and NK4E. They are better than FDA-approved drugs. We can give these compounds orally, and of course, the plan is to develop these cells. Again, I have this slide here just to emphasize. If you are trying to develop a drug or any product, you have to have what we call that's protection of your intellectual property. If you don't have the protection, you cannot be able to develop it. Because anybody can just come ahead and develop it and you don't have any rights. So you should make sure you have, these are the students, only students and postdocs that work on this project. These three are still in my lab. Sorry. So the last part of my talk is, the breast cancer, this project. And it starts out from all trans-retinoic acid, which all of us should know. And I can say 110% that if all of you can see this pointer, you have all trans-retinoic acid in your bodies. And you have all trans-retinoic acid, and the reason is because, the way we see, that's humans or animals, the way you are able to see involves, that's a retinoidal compound. When, that's light strikes on this double bond, there's a conformational change which sends that's the signals to our brains, and that's how we're able to see. So as I always joke, if you can see this pointer, you have all trans-retinoic acid in your body. All trans-retinoic acid has also been shown to inhibit that's some cancer. And as far as I know, it is an approved drug. That's for APL. And it's the only drug we know, which has been able to cure the disease. That's APL in patients. So you can see with the clinical trials done, within 40 to 60 days, that's some patients were actually cured of APL. That's the only drug we know. And it's also been shown to be implicated in breast, prostate cancer, and in other diseases. So that's why we were interested in it. However, the problem with the drug is that if you give it to patients, after a while, the resistance, this mechanism, it's because there is a metabolism of this molecule, to other molecules. And the key enzyme involved, CYP, is 26. So what's the implication of that? It means that, of course, you have to increase the dose of ATRA to the patient. And then, of course, if you increase doses of drugs, and their side effects. So our idea was why not develop molecules which can inhibit this key enzyme involved in this. So which means that you can give a low dose of all-trans-retinoic acid, plus inhibitor. And as I said, we all have all-trans-retinoic acid in our bodies. So if you give this inhibitor also, you may increase the levels in our bodies, and that could be that's effective. That was our interest. Again, when you want to design these molecules, you have to base that actually on what you know. Again, the heme ion is involved. We now ask if you modify this key framework and attack a group at C4, that's around where this metabolism takes place, you are likely to inhibit the enzyme. And indeed, we're able to make molecules which do that. So this slide just in the synthetic schematic for one of our lead compounds, which I'm not going to spend time on. For the sake of time, we'll just go on. So with this slide, it just summarizes where we started out and what we ended up with. We also found out that these molecules, in addition to inhibiting CYP17, they also impact on the androgen receptor. They also impact on the MNK1 and 2 ERF4 pathway, which I've talked about. And these are some of the lead compounds that we had. So we discovered what you call the first, that's MNK1 and 2 molecular glues, that reduce the degradation of the MNK1 and 2, and start to block this pathway. So, for the biologists in this lab, in this group, that's the RAS, this PI3-AKT pathway mTOR. That's the RAS again, mTOR. It all goes down to I can't see. What if we move this a little bit up? Anyway, we're not saying that, let me just know this. So, alright, so if we inhibit that, then you can block this protein translational complex. So that's what happens. Okay, good idea. So you can just praise that up. A little bit. Yeah. So, for the biologist again, some of you must be asking, all of us need, that's protein synthesis in our bodies. All animals need protein synthesis. So, but here, we're talking of inhibiting. So the question that arises is, kill all cells. Because when you're inhibiting cells, you want to kill them. Kill all cells, and not just the cancer cells. So, the main reason is that the mRNA, and we have heard a lot about mRNA during COVID. There are two major groups. You have what you call the strong mRNAs, and you have the weak mRNAs. The strong ones are what we call the housekeeping genes, and the ones we need for development. These mRNAs are well translated, even when the EIF4E complex is limiting. However, in diseases and in most cancers, the weak mRNA are translated when the EIF4A complex is upregulated. They're translated when the EIF4A is upregulated. So if you inhibit the upregulated EIF4A, you're able to inhibit the cell proliferation, suppress angiogenesis, repress the metastasis, and increase that cell death, that's apoptosis. So it means that if we're able to reduce EIF4A complex, we would have these minimal effects on the total protein synthesis. In another sense, I can explain this also through this graph. In normal growths, you can see that if you look at the rate of protein synthesis, when the EIF4A is limited, you can see that the housekeeping genes are all formed. That's a plateau. But in diseases and most tumors and metastasis, you have an increase in EIF4A, which binds to this complex. You can see these mRNAs are forming, and which go on to form the oncogene. So if you inhibit this, say to this level, you inhibit the cancer cells, but the normal cells, would stay alive. Just the same thing I said, you reduce EIF4A, and you have this minimal effects on the normal protein synthesis, and therefore minimal effects. I'll end up, of course, with breast cancer, and in this project, we're focusing on breast cancer that's triple negative. Now, there's a new subclass for triple negative breast cancer, and I will explain that to you in a minute. It is known that in our lifetimes, 1 in every 8 women would be diagnosed with breast cancer. There are 3 major causes: the ER positive, the HER2 positive, and you have drugs for this, specific drugs for these types of breast cancer. However, with the triple negative breast cancer, there's no specific drug approved for those. However, we also know that of this triple negative breast cancer, that a high percentage of them do not possess the androgen receptor. So those are called the QNBCs. That's for triple negative breast cancer. It's negative because it does not express the estrogen receptor, the HER2, and the progesterone. And now, this percentage do not express the AR. And if you look at it in this curve, the patients that have this low expression of EIF4A, the prognosis is quite good. However, the patients that have a high level of EIF4A, the prognosis is very poor. And in patient samples, you can see the ones that are cancerous, you can see they express the EIF4A. Why those that are not cancerous have minimal these levels of EIF4A. And it's been shown that in most cancer cells, they express this EIF4A. Thanks. That will soon be done in a few slides. Very easily. Alright. So, with our molecules, in principle, they can inhibit all subtypes of breast cancer. The reason you look at this line, which has almost this all subtypes, which means it includes the ones that express the ER alpha and the HER2 lineage. And as you can see here from the Western blots, the EIF4E and the phosphorylated form are all upregulated. So in principle, because we are able to impact on the EIF4E, these molecules, can be developed for all classes of breast cancer. Again, I have this slide. A lot of the time when you talk about breast cancer, you think it will only affect females. Okay, that's 2% of all breast cancers are found in men, have been identified in men. So you should know that men can also have breast cancer. So I already mentioned these, the first MNK degrading agents that were identified. And you can see in this slide, that's the MNK1. With our molecules, you can see the MNK is down. And the active form of EIF4E, the phosphorylated form, you can see the phosphor is down. These two molecules inhibit just the enzyme activity of MNK1 and 2. And you can see the MNK1 and 2 are still there with these molecules, but the phosphor EIF4E is down. But our molecules are able to induce the degradation of MNK1 and 2, and as such, will prevent the phosphorylation of EIF4E. That's the major difference with our compound. So I'll just go quickly to the in vivo studies that we first did. Usually, you can see here, I say, IP dosing. When you make a new molecule, to give it, that's the best chance. That's why you are testing it in animal models. You want to have many sites to the mice in a way in which you make sure it gets into the system. And the easiest way for any drug to get into the system, say in mice, is through the IP route. So that's what you want to try first. That's to give that's your new molecule any chance of whether it's effective in animal models. So this study was an IP study, and you can see, that's the control of the tumors when there's no treatment. And with our different molecules, you can see that they inhibit the tumors very well. These are examples of some mice. Again, we take, the body weights. And at the end of the experiment, we excise out the lungs, the liver and the kidney, and check to see, whether your molecule is not toxic. That's important. And as you can see, there's no difference with our molecules compared to the vehicle. An important thing also you want to check out is that although you are inhibiting tumor growth, you ask yourself, what is it doing to the target that you are after? So if you can look at this now, this is the Western blot of the tumors we have excised. You can see the MNK1 and 2 are down, which we showed in vitro. The phosphor EIF4E is down. Our molecules do not impact on EIF4E itself. The cyclin D1 is down, which we expect. The BCL2 apoptotic is down, and the BAX, this anti-apoptotic, is up. So you can see if you compare the BCL2 BAX, we are able to show that that is increased. Usually, that's the cancer cells. One of the big characteristics is that they refuse to die. So what you want to do is to have drugs we can use that cell death, that apoptosis. And as you can see here, we have been able to show that we increase that apoptosis by, you see the BAX is up and the BCL2 is down. That's an indication that is causing that cell death via apoptosis. Alright, so we went ahead. As I told you, usually you try the IP route first. since we have shown that the molecules work via IP route, and we will now compare the IP versus oral. Usually, with most drugs for compliance, you want to go the oral route. It's easier for your patient to take the drug oral. So you want to make sure, you want to ask the question whether you can give the drug oral. And in this case, we compared oral and IP, and you can see, there's not much difference. So here you can see I have this structure. This, okay, that's for the chemist. You have here, okay, that's the C4. What is the C4 group called? That's the chiral center, which means you can have the E1 and E2. Okay, it's the non-tumors. We're able to show here that there's not much difference, okay, between the E1 and E2 and also the racemate in our study. And then on this slide, of course, we looked at, that's all the tumors, and you can see in some cases that the tumors are being eradicated. And again, we'll do the Western blots also to show what's happening. So this slide is a summary of that's a triple-negative breast cancer cell. Tumor. Okay, that was coming from a patient in our cancer center hospital. And you can remove the tumor and then grow it in mice. So I wanted to ask the question, whether this tumor. Okay, that's from a patient. Okay, that's patient derived. The other ones I showed you, they are tumors which you develop from cell cultures. But this one, it's a direct tumor from a patient in our this cancer center. And as you can see again, these tumors are inhibited. Now if you look at, again, we looked at the MNK1, you can see it's down in the various tumors and also the first. AIF4E. The cliff bar is increased, which shows you that the cells are dying via apoptosis. Alright, I had mentioned that what kills the patient is when the tumors spread to other organs. You want to ask the question, can their molecules also inhibit the migration invasion of tumors? And we show here that yes, our molecules can do that. In vivo, we inject the cells, these fluorescent cells into mice. You monitor them for days. As you can see in the control, you have this now, it's metastasized to the lung area, while in the ones that we treated, it was just one mice actually that had. That's the metastasis. So our molecules are able to inhibit. That's not just the tumor growth, but they also are able to inhibit. That's metastasis of the tumors. So this is a summary of the studies. We went ahead and made some molecules. That's with deuterium. I'll just keep a number of slides for the rationale, for the sake of time. Just to show you, that we're able to improve the efficacy of our molecules. So this slide here shows the effects. On tumors. This derives from white patients, Caucasian Americans, and that's from black patients. That's the control. You can see the tumors are growing. That's the lead compound, the VNLZ152R. When we now have molecules, that's the deuterium, we are able to improve, that's the efficacy, as you can see in these tumor models. That's regression of the tumors. So these molecules, again, they've shown can work, that's with white patients and also blood patients. Again, we showed this, that's the models that do not express the androgen receptor. Here we compared it, with FDA approved drugs, and our molecules, are better. And that's the waterfall plot again. That is the impact. We're comparing, that's the parent compound shown here. And when we have this molecule, so that's the titration, you can see the impact. There's still more regression right there. Again, this slide just summarizes, that's what I just told you. And we have our lead compounds protected. That's their patents. These are the students and postdocs who worked on this project. We have now formed, that's a company. That's Isoprene Pharmaceuticals. So this is a company formed by the university, and I own part of the company. You may want to ask the question, how would I still be a university professor and I'm a president of also a company? What explains it is that the university has, what we call management plans, conflicts of interest management plan. And the university is involved, to make sure, that your work as a professor in the university does not impact on your role in the company. So you have the conflict of management plan. And that is approved by your head of department first, by your dean, and then we have the university president. Here, it is the vice chancellor who has to approve that conflicts of interest management. And that's monitored here, and so you can form companies. So we have the grants from the NCI. That's a small company now, has this grant. And we are collaborating with the CRO, that was pharmaceuticals, to develop these molecules. These are the aims we have for this project. It's a 2-year project. Because of my involvement in the company, I got this award. That's in 2021. That's Entrepreneur of the Year. The Grant Entrepreneur of the Year. This is our format. That's the nominator. For the award. You have to send out to assessors. Whether you deserve that. So these were 2 of the people external from the university that assessed me. And internally, the department chair is involved. The cancer center is also involved. And the CBT director, were all those that supported my nomination. This is the awarder. He has the final say. That's the vice chancellor of the Njar case. In our case, we call it the university president. So you can call it a vice chancellor, this AKA president of ACD University. So that is, I will acknowledge, my wife, could not come. She's in Abuja right now. And that's my son also. He could not be here. I acknowledge, that's the funders of my research. This slide, is important because if you don't have funds, you cannot do any research. And I would also like to point out, we are mentioning here, okay, the NIH, the National Institute of Health, the NCI, the National Cancer Institute. All these institutions are the government owned. And so the government, of Nigeria, should support that basic and translational research. You get, very few funding or very small funding from industry. I was supported, by Togan. Okay, but if you add, that's the percentage of the funds that I've received over the years. From the private company, less than 1% my funding. So the government of any country is very important. That's for the development of research, that's in academia, the translation. I'd like to thank all of you for listening. And I thank the leadership of the University and also your Chancellor, who was the brain behind me being here today. Thank you very much. And I'll be glad to answer any questions.
Prof, please kindly come out, please. The student colleagues, please let me know. Let's do this, a stand ovation. Chancellor, sir, with you colleagues, we have listened to the student scholars, and that lecture has been highly impactful, insightful, and we have some takeaways from the lecture. Maybe before, if there are any questions, raise up your hand, and I would like, yes.
Good afternoon, sir. Good afternoon, everyone. My name is Dr. Oniye Adesola from Medical Laboratory Science Department. I want to really thank you for this wonderful presentation. I see a lot of specialty has actually gone into this, and it's mind-blowing. Congratulations, sir. Okay, I have two questions I want to ask regarding the area of prostate cancer. You mentioned the analog of, the anti-androgen, galactron. Okay. I'm thinking, I don't know, if galactron has been tested on patients that have prostate cancer, high PSA, but has not gone through the stage of metastasis, talk less of having castrate-resistant prostate cancer, perhaps it could help their situation so that they won't go into the metastasis stage, since it's effective in other stages aside the stage 3, probably it could help in that area. And then the second question is, when we are developing drugs for commercial purpose, I think it should still be better than the previous FDA-approved drugs. Yeah. So I've learned that the process of using agonists or antagonists of androgen and even bilateral orchidectomy, that is the surgical castration, has led to metabolic alterations in patients with prostate cancer, which led to alteration in lipid profiles that has predisposed them to cardiovascular risk. So sometimes even after the treatment, some of them die of other causes aside the prostate cancer itself. So they may die of cardiovascular issues. So even after the treatments, mortality rates of prostate cancer is still high after castration. So I'm thinking for the commercial drug or this anti-androgen developed, if it could address the issue of metabolic alterations without altering metabolic profiles, so that this patient don't go into the stage of developing cardiovascular risk factors. Thank you, sir.
Take one questions. Professor.
Thank you very much, sir. It's been very, very interesting. My question is this, sir. How many pharmacists do you employ or invite this kind of research? I know you're a pharmacologist, and of course, this research you've been doing is being multifaceted. How much of the input of pharmacists was in your research, if you had any?
Okay, Prof, I think okay, Professor Budalo, third question.
Thank you very much, sir. Thanks to our guest lecturer. It has been with excitement that we have gathered here today, and the lecture has been eye-opening. I personally have a lot of questions, but to avoid it sounding as jargons in the eyes of many here, or in the ears of many, I think I will see you personally, sir. I have a lot to learn from you.
Okay, all right.
Yes, sir. However, I do have a few questions. My first question is in line with what Dr. Njar raised. Sir, is there a possibility of BPH, BPH, growing into prostate cancer? If that possibility exists, why didn't we look at the possibility of targeting 5-alpha reductase during your study, sir? Our findings from that experiment to address the issue that she raised. Before we start seeing the possibility of it spreading from prostate to other organs, we know that the molecule galaterol is able to arrest it at that stage before BPH, before become prostate cancer. That is if that possibility do exist. Secondly, sir, the idea of using loss in body weight as a marker, okay, to determine the toxicity of our drug, is it really reliable? Knowing whether some drugs may express their toxicity without interfering with body weight. Again, for the aspect of breast cancer, sir, the in vivo study, the animals that were used, I think rats, right? The rats and mice. Were they genetically modified, or the breast cancer was induced? And if the breast cancer was induced, what is the agent that was used to induce that breast cancer? As I said, there are a number of questions here. Let me just leave this to me. And I'm grateful that I am here today. All right, thank you, sir.
All these, all these are very good questions. I like to stand up. So all these are very good questions. So let me take the first question. She asks whether we're able to test our lead molecule. That's on patients who are not like resistant. Look at that castration resistant patient. We have not done that yet, but we have shown, in the laboratory, that if you inject the prostate cancer cells into mice, for them to develop tumors, but if, within the next day or two after, this inoculation, if you start treating the mice, you do not get any tumor growth. So I believe that, these molecules, will also work in that patients who, the prostate cancer, is not yet resistant. I think it will work. In that sense, there are a number of drugs actually used, in such patients. The challenge is with the patients who have become like I say, this resistant. So that's why we're actually focusing on the castration-resistant patients, for now. You also asked about the side effects of these drugs, resistance. Well, what I would say to you is that there is no drug on X which doesn't have side effects. So if you are just focused on, because there are side effects with some drugs, then you will not be able to develop any drug. So again, when you administer drugs to patients, you monitor them, to see the sides, the potential side effects or side effects that will come up. You start treating the side effects. Say, that's the first question. The second question he asked the input of, that's the pharmacist. Yeah, you asked about the input. So, drug discovery, per se, the early stage of drug discovery does not involve that's pharmacists at all. Remember, what you are looking out in developing drugs. You want to establish a dose. The dose of the drug is not the role of a pharmacist to look at the dose of the drugs. And the PK studies, pharmacokinetics, there are experts that are involved in pharmacokinetic studies. They are not pharmacists at all. So, as a pharmacist per se, does not really have a role in the development of drugs at all. If you just talk about pharmacists, it's in the end actually when there's a drug approved, okay, that's when, I mean, now get the pharmacist, this involved. Okay, but remember, at the Food and Drug, Administration, where, drugs are approved, I mean, there are pharmacists, of course, involved there. I mean, who also look at, that's the PK study, the toxicity studies, things like that. But they are experts. I mean, they are not just the regular, that's pharmacists. That's with the, with the first degree in pharmacy. And the third question, the BPH. Look at that benign prostatic hyperplasia. I mean, that's a big word, BPH. I mean, to simplify it, it is, that's an enlargement, of the prostate gland. I mean, that's what that is. And you should be assured, as far as I know, that BPH, does not transform, that's to prostate cancer at all. There's no evidence, that BPH gets, that's transformed to prostate cancer. I've had that argument actually before. As far as I know, there's no evidence of that at all. I do know, of course, that you can inhibit the enzyme, 5-alpha reductase, which is, they are, that's approved drug for that. That's Propecia, which, for example, is one of them. It's a 5-alpha reductase inhibitor. But it's, it had been tested a long time ago for prevention, of prostate cancer. That was a long study done by the NCI. But the results, were very mixed. So as far as I'm concerned, there's no 5-alpha reductase inhibitor that is approved for the treatment of prostate cancer. They are approved for the treatment of BPH. So you talked about using the body weight as a test for toxicity. Remember I said that, that is, the earliest test, we use. Other than that, if you remember, there was a slide I showed, that's what we looked at. That's the liver, the kidney, and the lungs. You also look at all those. That's for toxicity. If, any drug is toxic, when you examine these key organs, you can compare them with the control. You would be able to tell whether, the drug, could be toxic. But the easiest way, as far as I know, if you just want to initially test, that's for toxicity, look at the body weight loss. Then, the breast cancer model, you asked, so, what we typically do, you take normal cells, of breast cancer, and inject, that's subcutaneously, it's under the mice. And after, usually about 2 to 3 weeks, they will likely start growing, that's into tumor forms. And it's only when they start forming tumors, you would now be able to measure them, the sizes, with the caliber. And based actually on the measurements, you are able to calculate the volume. I mean, so the graphs you saw there, a lot of the graphs you saw for the anti-tumor studies, and we're actually plotting that's the volume of the tumor. I mean, from when you started the experiment, I mean, to when you end the experiment. Okay, at the end of the experiment, we will also excise these tumors. And you can see the photograph that I showed of the tumors. That's at the end of the experiment. And I will also use some pieces of those tumors, I mean, to check, okay, that's what the biomarkers that I talked about, the MNK interplay. The mice that we use, they are immune, okay, that's compromised. Okay, but there are also, I mean, some experiments, like, okay, there are some strain of experiments where you can use, okay, that's normal mice. Okay, for the most part, I mean, the mice are immunocompromised. Okay, but they are proved likely, I mean, for such studies. I hope I've answered all your questions. Thank you.
Emeritus Professor Oladeko. That's our pioneer vice chancellor. Please let's give him a round of applause.
I'm very intrigued by the presentation. I really admire. I'm very intrigued by the presentation, and I really admire the very things that are common. I think only those with chemical background in chemistry or in medicine, be able to appreciate the complexities of the various things that are coming. But I think what would be of interest to us generally is normally published that BPH, prostate cancer, and things of that nature, breast cancer, tend to be featured in people of Negro origin. Is there any, this should be of interest to all of us all. Is there anything that you have that can give us, throw a light on that, those people of makeup, is there any, what I can, genetic factors that makes black people tend to have prostate cancer? Quite a number of prominent blacks in this country, for example, have passed away because of prostate cancer. Is it in any way linked with the origin of inheritance factors or genetic considerations which makes black people tend to have these features than those people who are of other races, say in Europe and America? All right. Sorry.
Oh, thank you for that. That's a very good question. So, I mean, what we currently know is that most cancers could be either because of our genes. Okay, that's genetic. The environment, I mean, also plays a role. And for example, the foods that we eat, it also take a role, I mean, play a role. It has been shown that, people, okay, that's from Asia, when they now migrate, okay, that's to the Western world, their incidence, I mean, of prostate cancer increases. I mean, in that sense, I mean, that would actually tell you that, okay, that the diet, I mean, would have changed. And that, I mean, is having, you know, an impact on the disease. It's known, okay, that, I mean, some plaques, okay, like in the Caribbean, like I mentioned, that's where you have the highest rates of, okay, that's prostate cancer in the world, and even the death rate. And that's has been associated, I mean, with slave trade. It is, you know, being discussed that, you know, because, I mean, of the hardship, I mean, of the blacks, okay, that during slave trade, there is an upregulation of how are they actually able to produce, okay, that's the androgens. And it looks like, I mean, the levels, I mean, of androgens, okay, that's in blacks of those areas, is much, you know, enhanced. You know, remember I told you that it's been shown that these androgens, you know, will actually stimulate, okay, the growth of prostate cancer. So, to me, okay, that seems to make some sense. Why, I mean, of course, you have it elevated in blacks. In the United States, for example, another issue is that all the clinical trials, or most clinical trials of drugs with prostate cancer, they were actually developed with, they were actually developed that with patients, I mean, from the white population. Okay, the blacks, for example, in America, I mean, do not want to get involved with that in clinical trials. I mean, the enrollment of blacks in clinical trials in the United States, for example, I mean, is very low as compared to the whites. I mean, so that's why, of course, I mean, you will not know, I mean, the differences that are involved. Okay, but that's, okay, being addressed now, where, I mean, in research, I mean, we are told we should include, like what you call the models, I mean, from both, you know, groups, okay, that's blacks and whites. I mean, so the hope is that, I mean, if, I mean, they are involved more, okay, that's in clinical trials, you may be able to narrow the gap, so that disparity actually of having these drugs, you know, and the impact. So the NIH, you know, has, okay, now they actually have a center which looks, okay, at disparities of, I mean, of how drugs are developed currently in the United States. And we are hoping, I mean, of course, that gap, you know, between the white patients, I mean, the white population, you know, the black population, I mean, will be closed. Thank you.
We anyways, 2 more questions. Then after that, the Chancellor is going to wrap it up. Yeah. Okay, Provost.
Mr. Lecturer.
Thank you.
Let me first dig a foundation to my question. Some while back, I had a patient, 82 years old, who was complaining of what I suspected to be diabetes mellitus. And so we did a test. It was positive. And then, as a young doctor, I sat him down to give him advice, nutritional advice. 82-year-old Egyptian man. Don't take gari, don't take fufu, don't take rice, don't take. Immediately I touched pounded yam, he blew up. Yes. He said, look, I am staying alive not because I enjoy to go to parties. I am staying alive not because I enjoy drinking. I am staying alive to eat pounded yam. Let's draw a parallel between that and the 60, 70, 80-year-olds with prostate cancer. You've been talking about castration, surgical castration, chemical castration, castration on all forms. Can we look at treatment of cancer of the prostate from another perspective? Because some, even at that age, still do enjoy sex. It's part of the human nature, part of the human drives, hunger drive, sex drive. But by the time you castrate him, you have virtually, indirectly killed him. So why is he staying alive? I'm now putting it to you, sir, that you look at other enzymes, other methods, other receptors, apart from androgens. Thank you.
Professor Adino, do you want to ask a question?
Yes, sir.
Okay.
I just want a distinguished lecturer to give us information how he was able to command cooperation from different departments like histology, pharmacology, and so on.
Okay. And after that?
Yeah. Starting from the I just want to know, I got to know my bigger brother about 2 months ago because of this prostate problem. And personally, I'm now using yoga and aminosin. Yogurt. Yeah. Recommended by Professor Oye, yogurt and so I don't know the implication. Because the doctor said that every drug can be taken as I don't want to die young. That's not my
All right. Thank you, sir. My first question is about the disparity between men and women. 1 of 8 women is usually diagnosed of cancer, only 2% of men are. So I'd like to know what is responsible for this. And secondly, cholesterol in Nigeria is badly maligned. So I think that the scientists don't have agreement on the significance or the importance of cholesterol. And the fourth one has been answered. Lastly, I'd like to know, you said that cancer, what kills the patient is when the cancer spreads through the cells. So the question is, getting the cancer or the cancerous cell at the initial stage is the only single most effective way of addressing cancer. Is this true, sir? Thank you.
All right. I hope I remember all the questions. So the first question was that, okay, that's patients who've been focusing on okay, that's trying to prevent the action, of androgen genes. That's via, that's removing the testes. Okay, that's castration. So what I said was yes. That's Charles Huggins. Okay, that's won the Nobel Prize. For that discovery. Okay, but our idea now of inhibiting an enzyme, that is the same thing, has an advantage. One, you'll be able to inhibit that's androgens. Okay, that's from all sources. Remember, that's the castration, and it will only eliminate the production of androgens from just the testes. So, with our CYP17 inhibitor idea or technology, you can inhibit from all sources. Okay, but another advantage is that, okay, let's say, you are taking the drug that inhibits CYP17, and you start having complications. What the doctor can do is to reduce the dose of the drug, right? And if it's still that difficult, you can actually stop that particular therapy. It's a big advantage, okay, that's over when you remove, okay, that testes. You cannot replace these testes. So, and again, as I said, there's no drug that you take that doesn't have any side effects. All you want to do, to be realistic, you should just monitor. And it could vary, okay, that's from patient to patient. So it's not as if, if the same thing is happening to patient A, it's exactly the same thing that happens to patient B. No. Our enzyme systems are quite different, things like that. So those are things that, you want to watch. The third question was, the gentleman out there asked, how was I able to get collaboration from the different disciplines? All right, so I will actually start out. Okay, that's from what I did. You can see in my talk, I've talked about, okay, that's the chemistry. Which, I'm well trained for. But in my laboratory, I am able, to incorporate, okay, the cancer biology, the pharmacology, and all that. So I think it would be, in my view, okay, it's useful to hear, to hear this story. So, as a chemist, I've always been interested, okay, that's in drug discovery and development. in chemistry and organic chemistry and synthesis, I mean, you have to design molecules. You have to test molecules. You have to have, okay, that's a target. Okay, but as you say, I mean, there are different disciplines. I mean, so how do you do it? You would, okay, that's collaborate with other, okay, experts. Okay, but earlier on in my career, I found out that collaboration actually sometimes like slows down things. So in 1996, I now wanted to start learning, okay, that's some biology, okay, some pharmacology and things like that. So I asked myself the question, what would be the best way to do it? I now decided to collaborate with an expert, okay, that's in cancer biology and pharmacology. Okay, that's Dr. Brody in Maryland. I mean, so when I told my chemistry, okay, colleagues that I was going to Maryland, I mean, to collaborate with Dr. Brody. I mean, so the big question is, well, she's not a chemist. I mean, why are you going to collaborate? Why are you going to spend time in Harvard? Okay, so my answer to them was, I'm not going there to learn chemistry. I mean, I knew, okay, that's enough chemistry. What I was going there, okay, was to learn the biology and the pharmacology. Okay, and that's what I did. And that's what I did. So, in fact, today, I would say, I've graduated, okay, more PhDs. I mean, in pharmacology, in cancer biology, and in chemistry. So, it's all what, I mean, you really want to do. I mean, you mentioned, I mean, all these other disciplines. Yes, okay, that's most of the best things. Okay, there are centers actually that specialize, I mean, in all those techniques. I mean, so all you need to do is, I mean, if you decide now you want to do this test or whatever, I mean, you have to collaborate with those labs and do this. Okay, but in my case, of course, because I'm able, in my lab, I mean, we design, okay, that's a molecule. In my lab, I mean, we're able to test these molecules in vitro. And I also decided that it's not just enough testing things in vitro. You want to see, you know, what they do in the animal models. Okay, my lab, you know, also developed, okay, that's all these techniques. You know, so we do, I mean, our animal studies. Yeah, things like that. I mean, it can be done. Okay, but I will tell you, I mean, another thing I know. It's easier if you have, okay, like me, the background, okay, that's in chemistry. I mean, to now incorporate, okay, that biology, this cancer biology and all in your research. But not, okay, that's the other way around. So in fact, I don't think I've seen anybody, and I would say that in the world, who trained as that's a biologist or pharmacologist who is able to incorporate okay, that's chemistry in his lab effectively. I have not seen. It's easier, I mean, for a chemist, somebody trained in, I mean, organic synthesis or whatever, organic chemistry, I mean, to incorporate, I mean, the biology and the pharmacology. And back, at my A levels, and I said to myself, the grade that I got in biology was better than my grade in chemistry. So I told myself I'll be able to learn the biology, and that's what I did. Thank you.
Please, can we give a round of applause? Now, I would like to call on our eminent and invaluable Chancellor for his own contribution. Let's give our Chancellor a round of applause. He is the person that translated this program. Already, we are having a collaboration with the Chancellor, sir.
The Vice Chancellor, very distinguished guest lecturer, Deputy Vice Chancellor Registrar, the librarian, well, what can I call this? Very, very distinguished audience. I think I am quite happy and privileged to be able to respond to some of this. I'm a chemist, so if anybody is to review this lecture, I'm in a position to review it. So that is one. And Professor Njar has just told you that it's easier for a chemist to learn biology than a biologist to learn chemistry. That just shows you how interdisciplinary chemistry is. That sum it up. And I used to wonder, God created Earth and heaven. Is he a chemist or a biologist? I mean, those are fundamental questions to ask. And the greatest structures that we have today, the structures in biology, which arises from evolution, that it has all been designed. And when God created us and said, let's make man in our own image, he wanted us to be co-creators with him. That is to continue where he started, not where he has stopped. So these are fundamental issues that I think we have been exposed to today. So let me start. Why is today one of my happiest? One, I think the greatest achievement or honor that the professor can attain is when he sits down and is lectured by his own students. Thank you, Professor Njar. I think I have earned it. And I'm glad to share that honor with all of you in listening to this beautiful lecture. Two, when I walk into my lectures, my students used to laugh because I greet them, dear colleagues, and they would all laugh. Aren't they my colleagues? Aren't they my superior colleagues? So if you have not produced your colleagues or your superior colleagues, then you have not taught. So that's what we really have been educated today. And I think that's it. Take home. I always look at my student, and anywhere, even if I'm going on the road and my student says, excuse me, sir, I will just stop. It's almost like a command. When he says, excuse me, sir, I will stop. Listen If it's something I can answer there and then, I will do. If it's something I needed more information, I said, can we talk about this later? But suppose I had refused to teach Professor Njar. Look at the waste I will have caused to a nation and to the world. I'm just trying to draw this so that we can have a lot to take from. Professor Njar was just, I just heard that he was going to be around, and I then said, how about giving a lecture to us while you are here and all the like. Then we fixed the date, and Dr. Ladu discovered that in actual fact, it fell during our vacation. And this lecture was not going to hold because we were still on vacation. But we consider it very important that the university should not be deprived of this opportunity. And I think that Radio Congress, Dr. Woolard and others make sure that this assembly happens. There we are, listening to what I will describe a fantastic lecture. And now I like giving some of my own experience. I've taught and researched at many centers in the world. But there is one university where I was part of the research team, and I will let out a secret to you. In that university, Nobelists abound. If you don't run into a Nobel Prize winner in an elevator when it's going up, you give it to one when it's coming down. What is the secret? I learned it. And I was taught that what they do, they invite all authorities, including their competitors across the globe, to come and give lectures. That's how they were able to do far more Nobel Prize winners. And they will do everything possible to make sure it's important. And we were taught, I was part of the team, that once you listen to that lecture, within 72 hours of that lecture, you must gather and reflect on that lecture and see in what way it can help your own research, or in what way you can modify that work to be able to improve your research. I hope we will do that. And I hope that it will also teach us that the university will continue to bring excellent speakers all over the world, because we want to keep the university of the highest standard. And I think we're setting the tradition, and that we listen to this lecture, and that within the week there will be groups and teams getting together, discussing as many of the questions as you have asked. Why are there genetic differences in BP and so on and so forth? If you do ablation, are you killing somebody or making him alive and things like that? Or what are these are things that come from lectures. And it also builds you up. And of course, let me also remind all of you, why are you a lecturer? I think I discovered that many years ago. Lecturers are perpetual students who have stopped paying fees. Vice Chancellor, did any of the people here pay fees? No. And yet acquiring an education. So let's make this an intellectual center, and let us make sure we incorporate a culture of learning. And I'm a chemist, and I learned a lot of biology from my friend Professor Son, with whom we worked together in the story on immunity. And I found as a chemist that in fact to use chemistry to understand biology is the greatest goal. So thank you very much for this beautiful lecture, and I hope that arising from the I've engaged Professor Njar, how much we can collaborate, and he has given me a positive response, how much we can be even willing to give an online lecture. As we designed a course in medicinal chemistry. He doesn't need to fly from Maryland. You can give it here and we will listen. And that the teachers here will examine. He doesn't need to come and do that and things like that. And that he's also willing to share, and I think we learn experiences about doing basic chemistry, applied chemistry, running into entrepreneurship, and that the university also has learned that we should set up a patent board, encouraging people to be able to set up companies without conflict of interest, and the university will also build. So those are things I thought I have learned, and as a takeaway from here. But you have gladdened my heart. You have provided a fantastic audience to a very beautiful lecture. Thank you, Mr. Njar. I think that if we are beginning the year like this, let's see many more of it. I think that the sky is the limit for Lead City University. And when you get there, we will remove the sky.
Please, can we once again appreciate our council, the Chancellor of the Pilot. The Chancellor of the Pilot, and we pray that our good Lord will continue to strengthen you, grant you standards, and more prosperity. On this note, I would like to call on the registrar, that's Dr. Ebola, to give vote of thanks. Let's appreciate it.
Thank you very much, sir. I want to first appreciate the Chancellor again for bringing this first distinguished lecture to Lead City University. Thank you so much, sir. It's a very good way to start the year, indeed. And I know that this year, greater things, that will celebrate and bring Lead City University to that global realm is going to happen. So thank you, sir. We appreciate you, sir. And so, our first distinguished lecturer, we can't thank you enough. Thank you for bringing this very insightful lecture to Lead City University. Your wealth of experience, cannot be bought with money. So we appreciate you, and we would like to decorate you as a friend of the university. I have the university lapel with me, and I would like to put it on you as a friend of our university. Thank you. This will be a beginning of a very useful collaboration with our own university in Nigeria. So thank you.
Thank you very much.